Hello, and a very warm welcome to everyone joining us for today's Select Science webinar, Scaling 3D Cell Experiments with Confidence with the new Spark Cyto 3D AI analysis tools coming soon and start-to-end workflow automation. My name is Dora Wells and I'll be moderating today's presentation. So I'm delighted to be joined by Roman Petrovsky, Product Manager of Cell Imaging at TECAN, and Linda O'Leary, Manager Marketing Application Detection at TECAN. After the presentations, we'll move on to our question and answer session. Please feel free to ask any questions for the Q&A session at any time during the webinar. You can submit your question at the left of your screen. Just click on the purple tab. And now, without further delay, I'd like to hand it over to Roman and Linda for today's presentation, and I'd like to thank them for presenting to us today. Please go ahead.
So thank you very much for the introduction. I'm going to start with a brief introduction to the world of 3D. Then I'll be discussing the promise of 3D cell models, but also the challenges associated with them. Then I will hand over to my colleague Roman to tell you about the new Spark Cyto 3D AI software tools and how we at TECAN are enabling whole workflow solutions. Then we'll wrap up with a summary and a Q&A. So let's get started. A common starting point for molecular and cell biology-based research is 2D cell cultures using cell lines. For more than a century, it has been a foundational and very powerful method to help scientists answer biological questions, in part due to the high experimental flexibility, but also the reproducibility and ease of handling. However, while vital in making many groundbreaking discoveries, there are also significant failures, as findings in 2D often fail to translate to what happens in the body. As you can see here, there is a large gap between cells grown on a 2D plastic or glass layer and animal or human models. This is not only due to being isolated from a system, but also the manner in which cells grow, flat and in 2D. Cells grown in 3D, such as spheroids and organoids, allow for greater interactions not just with other cells, but also the environment and extracellular matrix. They experience a gradient formation, meaning nutrients, oxygen, and also drugs penetrate to different degrees. And this is particularly important when considering tumor response and in contrast to 2D cultures where all cells are exposed to the same factors and drugs. Given the right environment and signals, cells have the potential to differentiate into various cell types and form structures which mimic the organ or tissue. And adding 3D cell models into experimental workflows can bridge this gap and give more physiologically relevant insights early on. Let's take a closer look at 2D and 3D models. As I already mentioned, two major benefits of 2D cell culture models is the cost and ease of use. They are easily scalable and reproducible, as well as the wealth of data that already exists. However, 2D cell cultures fail to recapitulate the in vivo environment, resulting in cells having different morphology, different proliferation rates, and interactions than they would have in the body. It's also been widely demonstrated that gene expression profiles differ, and as a result of all these differences, they fail to recapitulate in vivo drug response. And of course, they cannot be used for personalized medicine. Spheroids are the next level in complexity, simple 3D structures usually generated from a single cell type. And this comes with a gentle learning curve with regards to handling of the cells and handling of spheroids and matrix. We can add in different cell types which are found in the microenvironment to add another layer of complexity. And while cells growing in 3D already bring us a little bit closer to the in vivo situation, there's still a lack of differentiation and polarization, so we do not see the types of structures that we do in vivo. But however, these deficiencies are addressed by organoids. And these are complex 3D in vitro structures derived from stem cells capable of self-organizing, self-renewing, and differentiating into multiple cell lineages. Organoids are more costly and require expert handling. However, the benefits are the gene expression profile is maintained. These models can mimic the organ from which they originate and recapitulate to some degree the function of that organ. Patient-derived organoids hold unique promise for personalized medicine, allowing clinicians to screen patient organoids which may have specific genetic drivers for a variety of therapeutics before giving them to the patient. And the need to fill this gap in physiological relevance can be clearly demonstrated by looking at the drug discovery funnel. Traditionally, at the mouth of this funnel, cell-based assays and 2D cell culture screens are heavily utilized. And with over 10,000 candidate drugs entering the funnel, there is a huge attrition rate over the 10+ years of the drug discovery process, which produces just one successful drug. With less than 10% of the anti-cancer drugs that enter clinical trials reaching the market. This high failure rate, not to mention considerable costs, can be attributed to inefficient preclinical models with low predictive power that simply cannot fully recapitulate the in vivo situation. However, organoids have been shown to accurately predict patient responses to targeted therapies and chemotherapies in the cases of colorectal cancer, lung cancer, and breast cancer, to name a few. And while drug response is critical to assess and predict, so too is drug-induced toxicity. And this is a major cause of drug withdrawal from the market. And here too, liver organoids have proven vital in predicting drug-induced liver injuries. And these promising results have also led to a change in legislation by the FDA, allowing alternatives to animal testing for purposes of drug and biological product applications. So 3D biology is not only changing the face of drug discovery, but also basic cancer research, immuno-oncology, and tumor invasion studies in the case of spheroids, for example, which are a little bit more simple cell models. And for the more complex organoid models, these are proving valuable in developmental biology, regenerative medicine, and disease modeling, to name a few. And as can be expected, with increased physiological relevance comes increased complexity. And this can be seen at the experimental level with long and tedious experiments and tricky handling of matrix, together with challenges in scalability and reproducibility. And in the case of organoids, this also comes with handling of sensitive or fragile cell populations, such as stem cells or patient-derived cells, long differentiation protocols coming with complexity and variability, and expert handling is needed. The learning curve here is much steeper from 2D cell culture. There are also some technical challenges associated with 3D experiments, such as complex formats. There may be objects in multiple planes within matrices. Imaging and analysis can be time-consuming, and complex shapes may be difficult to quantify. And as this area matures, researchers are now in need of scaling up their 3D experiments, enhancing standardization and reproducibility. And now I'm going to hand you over to my colleague Roman to tell you how we at TECAN can give you the tools to do this.
Thank you, Linda. Now we're coming to the star of the show, the Spark Cyto. The Spark Cyto is a multimode reader, and as such can measure absorbance using high-speed monochromators, fluorescence with our fusion optics technology, and luminescence, particularly useful for cell titer glow assays, which are common as 3D cell culture endpoint assays. As a distinguishing feature to traditional multimode readers, it has bright field and fluorescence whole well imaging capabilities with 2, 4, and 10x objectives. The Spark Cyto also has an integrated lid lifter and incubation capabilities with temperature and gas control and evaporation protection using a humidity cassette, which makes it perfect for live cell imaging assays and kinetic workflows. As a last point, the Cyto has an intuitive and user-friendly software, the Spark Control. Now let's have a look at the imaging capabilities the Cyto supports. The first feature works for bright field images and measures the area of cells that covers the bottom of the well, and thereby is a measure of cell confluency. Next, we offer multicolor applications supporting apoptosis and transfection efficiency. Lastly, counting applications like nuclei counting, viability quantification based on staining, and AI-supported bright field label-free cell counting. With Spark Control 4.0, we now add new 3D analysis functions, including AI-based segmentation of single and multiple spheroids and organoids for bright field and fluorescence images. Z-stacking functionality with the ability to create total projections from the stack, and a new image-based autofocus to support imaging of complex formats, such as round bottom plates. So let's get into the details. A key feature that is particularly useful for quality control of 3D cell culture models is the ability to image, segment, and analyze 3D objects in bright field, removing the need to stain the objects. So what you see in this image is a set of dog liver organoids that have been segmented by our AI-based algorithm in bright field, which is visible through a yellow segmentation mask around them. Based on that mask, a set of key parameters is calculated that will give the scientist an understanding of the quality and developmental stage of the objects. These parameters are count, length and width, the area of the object, and eccentricity, which is a measure for circularity. So you will be able to track the development of your spheroids and organoids without the need for staining, thereby avoiding potential interference with their development. Fluorescence images can also be used for the AI-based segmentation with the same morphological parameters as output. In addition, the fluorescence intensity within the segmentation mask can be determined for each channel. So you have the flexibility to use any of the 4 channels as a basis for segmentation and quantify fluorescence and morphological parameters from all channels based on that mask. Now imagine a typical situation where multiple 3D objects have been growing in a matrix and they are not positioned in the same optical plane. For this scenario, the included Z-stacking functionality is particularly useful, allowing the acquisition of multiple images in the Z dimension. After acquisition, the in-focus information of each layer of the Z-stack will be projected into one image, a Z projection. This image is the basis for the segmentation of the algorithm. In this example, the left side shows a single image layer of the Z-stack, and several spheroids are out of focus since they are positioned above or below that layer. On the right side, you see the Z projection of that stack containing all the in-focus information from the layers of that stack, meaning the essential imagery information of the spheroids of all the images within the Z-stack. As you can see, spheroids that are out of focus in a single image and therefore not segmented by the AI, since they are actually positioned above or below, appear in focus in the projected image and segmented by the AI. This can also be appreciated in the object count resulting from the AI segmentation being much higher in the Z projected image. So this feature allows for automated Z-stacking and analysis of objects distributed in all 3 dimensions of a microwell. In addition, since you can decide to save only the Z projection, it allows to significantly reduce the space on the hard disk otherwise occupied by the Z-stack images. Now let me show you a couple of examples showcasing the segmentation performance of the AI algorithm. On the left side, you see an image of dog liver organoids, and on the right side, mouse liver organoids. As you can appreciate, the algorithm can detect objects that differ in size, are positioned close to each other, or are different in contrast. In this case, we see a sample of human colon organoids, with the segmentation being based on fluorescence staining. Also in this image, the AI algorithm is able to segment different sizes, organoids in close proximity, and even more complex shapes. Again, showcasing the flexibility of the algorithm. To support imaging and analysis of single spheroids and organoids, which are often cultured in U-shaped plates, we developed a new image-based autofocus and a separate algorithm that is specialized to detect a single object within the well and ignore potential secondary structures like aggregates. I hope with these examples I was able to give an impression of the versatility of the AI-based algorithms supporting single and multiple object recognition in bright field and fluorescence and being adaptable to a variety of morphologies. Coming to the next feature, the matrix view was developed to give the user a comprehensive overview over what is going on in the whole microplate, with the data being displayed in real time. As an example for a typical kinetic measurement, the image on the left side shows the matrix plate overview of a 96 well plate with a mini curve for each well representing the development of a specific parameter over time. In this case, the curve represents the fluorescence intensity measured within the bright field segmentation mask. Selecting one of the wells results in the display of a more detailed curve on the right, where also more parameters can be selected and overlaid. In the plate overview, a thumbnail for the recorded image can also be displayed for each well. Summing up, the matrix view allows the display of recorded data in real time for the whole plate, allowing for a better tracking and visualization of long-term measurements. To give the user an even better visualization of the spheroid and organoid development, the new Spark control software allows for the creation of time-lapse videos, which can be created after a completed kinetic measurement. In addition to flat bottom and U-shaped plates, we also tested for the imaging and AI-based analysis of special plates. One example is the Agrabel 800 from Stem Cell Technologies, which has the basic format of a 24-well plate, but each of the wells contains a high number of microwells, which allow for the growth of single spheroids and organoids within each microwell. As can be seen on the magnification on the right side, not only did the imaging succeed, but also the segmentation, as shown by the yellow segmentation mask. A second example are the GRI 3D plates from Sun Bioscience. In this case, we imaged the GRI 3D 96, a 96 plate format with a PEG-based hydrogel forming the microwell arrays within each well. We are happy that not only imaging but also AI-based segmentation works for these and other special plates, increasing plate format flexibility and allowing for easy upscaling of established procedures. As a multimode reader, the Spark Cyto features multiplexing of imaging with luminescence, absorption, and fluorescence intensity measurements, supporting multiple types of endpoint assays. One prominent endpoint assay in the 3D cell culture field is the cell title glow viability assay, which the Spark Cyto supports via its luminescence measurement capabilities. A second TECAN instrument, the D300E digital dispenser, can also prove useful for this application, since it features precise dispensation of small volumes down to 11 picoliters. Now we are coming to the solutions TECAN can offer to support and automate a typical 3D cell culture workflow. One key instrument here is the Fluent Automation Workstation. The Fluent has automation capabilities particularly useful for pipetting and integration of TECAN or third-party devices. It can be equipped with a HEPA hood to create a sterile environment, ensure environmental control via integration of automated incubators, integrate with the Spark Cyto to allow for multiplex analysis. Integration in this case is possible from the side, from behind, and from below. It can handle both tubes and microplates with a robotic gripper arm, automate routine cell culture work such as cell seeding, media exchange, and cell harvesting via its multiple or flexible channel arms, and integrate third-party devices such as centrifuges, imagers, or incubators via TECAN networks. Now, let's take a look at a typical workflow in 3D biology and find out how TECAN instruments can help to automate it. One would start with the induction of stem cells or a biopsy, followed by seeding the respective cells into the appropriate medium or matrix to start the generation of the 3D models. At this step, the Fluent can automate the seeding process and media handling. Next, the induced spheroid or organoid cultures will need to be placed into an incubator to grow to maturity. This step also can be automated with a Fluent integrated incubator. During this growth phase, the quality and maturity of the 3D models will need to be tested at regular times, ideally without much disturbance. For this task, the AI-based segmentation in bright field images with the Spark Cyto is a very useful solution, allowing analysis of key morphological parameters without the need for staining procedures. After the 3D models have reached the desired maturity, they might be treated with respective drugs, for instance, for drug screening procedures in cancer research. Since this can require very precise dose dispensation, also here the D300E can be a great addition to the workflow. To visualize the effect of the drug, for instance, if an anti-cancer drug was able to induce apoptosis throughout the tumoroid or just on the periphery, the 3D model can then be stained with a cellular dye or antibody-coupled molecular markers. Also, at this step, the Fluent might potentially automate the staining procedure. Finally, to visualize the effect of the drug treatment and staining, the Spark Cyto can step in with its bright field and fluorescence imaging and AI-based analysis. However, in addition, it can also perform luminescence, absorbance, or fluorescence-based endpoint measurements to get more data out of the sample. Once the measurement is going on, the analysis can already begin, giving the user real-life feedback on the analyzed sample. The typical challenges of this workflow are the length of the development of the 3D model, the lack of standardization, and the low reproducibility. By using an automated system like the Spark Cyto for 3D applications, particularly in concert with the Fluent and D300E, these issues can be ameliorated, resulting in several benefits. Higher standardization during the quality control and monitoring of 3D models, increased throughput, the ability to multiplex a variety of analysis functions, ease of use with the Spark Control software, and more reliable and reproducible results. Coming to a close, let me summarize the 3D features that will come with the new Spark Control 4.0 update. Bright field and fluorescence-based segmentation with 3D AI algorithms, automated Z-stacking and the creation of Z-projection images, real-time data display on live cell experiments for the whole microplate, flexibility and scalability of experiments with multiple supported plate formats, multiplexing with cell titer glow or other luminescence, fluorescence, or absorbance-based assays, and automation support of the whole workflow with the TECAN Fluent, Spark Cyto, and D300E. If we could raise your interest, feel free to visit our landing page with more information on the Spark Cyto, Fluent, and D300E and their functions within the 3D cell culture workflows, or tune in to expert talks and publications on 3D related topics. To finish, let me present you a recent video showcasing the new 3D features of Spark Control 4.0. Okay, thank you very much for your attention. Now, Linda and me are happy to address any questions you may have.
Great. Thank you for that interesting seminar, Roman and Linda. Now let's move straight on to the Q&A session of today's webinar. So the first question I've got for you here is this solution research use only?
So yes, this is a research use only product, the Spark Cyto.
Perfect, quick answer there. So the next question here is, quite often the spheroids or organoids are not round, but instead dissociate. For example, how well is your algorithm dealing with that?
Yeah, so sure. Spheroids and organoids pose their own difficulties in themselves. It's quite a difficult model. You have some spheroids, for example, that in the first 24 hours, they're quite loose. They don't condense until a bit later. So in these cases, when you have cells that are quite loose and not yet compact, there we can still segment them and also follow that through compaction. If that spheroid were to be treated and you would have a lot of debris, there we would not capture the debris within that. So that was something we also wanted to train very well on, that we captured these different phases and also different treatment phases of a spheroid and of an organoid as well.
Great, thank you very much for that one, Linda. So the next question I have from an attendee here is, does the segmentation of objects in special plates, for example, Agrawell, work for bright field images or only for fluorescence?
Yeah, so I can take that one. So with the kind of the emergence of these specialized plates with a lot of these microwells, the illumination can be quite different depending on how these specialized plates, these microwells, are formed. Some of them are round, some of them are in pyramids. And that's why we wanted to be quite flexible with being able to segment in both bright field and fluorescence. So if you have quite a difficult plate that perhaps there's a lot of different illumination artifacts, for example, you can choose to segment in fluorescence. So we found that in general, the bright field works really well. Maybe not at the very edges where perhaps your sample is not complete or there's a little bit of a half a well there. But this is also why we gave that flexibility to be able to segment in both bright field and fluorescence, so that if something is not performing as well as it should, then you can have another option as well.
Excellent. Thanks a lot for that one. So the next question I've got here from an attendee is, can I use an existing Spark Cyto with the new 3D features? Maybe Roman, this is a question for you.
Yeah. Seems to be lagging a bit, but I try. So you can upgrade existing Spark Cytos. That's not a problem. So for the details, I would advise to contact our sales representative. Then we can clarify that. But the option exists.
Great. Thanks a lot for that one. Yes, I can see your video is lagging slightly behind your audio, but your audio is coming through completely clearly. The next question I've got here is, can I use different detection modes together with imaging in one experiment? Linda, perhaps you can answer this one.
Yeah, sure. So it's not only just being able to do these different detection modes as well as imaging within the same instrument, but you can really set up a whole method with the imaging and the different detection modes within one experiment. So if you take the example that Roman showed, which is the cell titer glow assay, this is a common endpoint for a lot of spheroid growth assays or drug screening assays, for example. Now, you can not only do that within one experiment, but you can also, by means of the injector, set that you can inject the cell titer glow reagent. You can also do your shaking as part of that experiment, do your incubation, and do your readout. And all of that information and all of those results are then captured within the same workspace. So you have less handling, you have less transfer from instrument to instrument, and you have better reproducibility being able to do that.
Excellent answer. Thanks a lot for that one. So the next question I've got here is, how many images can be taken per Z-stack?
Yep. So you can take up to 50 images. And I should mention here also that you have the ability to reanalyze the Z-stack and then create different Z projection from that stack.
Great. Thanks a lot for that one. And the next question I've got here is, how does the system handle irregular shaped objects?
Sure. So because we also wanted to be able to handle organoids, and we know that they're not so nice and round like spheroids, there's a lot of different types of origins of organoids. You can have patient-derived organoids, lots of different origin of from different organs, and they have very different morphologies. Not only are they irregular shaped, but they can also be different in contrast. And we spent a lot of time and a lot of effort in training the algorithm to deal with these different scenarios. And I think you might have seen within the presentation, Roman also showed some nice images dealing with, for example, cystic organoids versus those ones that are a little bit more dense, but also showing how we're able to very well segment those very irregular shaped things as well. So, and that's also what we wanted to be able to do when being able to provide this software and analysis features also for organoids, not just for spheroids.
Excellent. Thanks a lot for that one. I think we've got time for just a few more questions. And so one of these is from an attendee who just missed the beginning of the webinar. So I was wondering, when will the new Spark Cyto 3D AI analysis tool become available?
Well, the answer to that is immediately. So you can already start talking to your sales reps about this, and they will have more information about that. If you already have a Spark Cyto system, you can also contact them about an upgrade. And if you're looking for a new Spark Cyto, then contact your local sales rep.
Perfect. Excellent. I think we just have time for just one more question. So that is someone saying really nice presentation. So thanks a lot, both of you. It seems like these capabilities will ultimately generate huge amounts of data. Have you made improvements to the data storage piece of the software?
Mm-hmm. So, of course, when you want to do a lot of Z-stacks, of course, that means more data. So one feature that we did here is because we are analyzing the projected image. So you do have an option that you can immediately actually disregard or delete your individual Z-stack images if you don't want to maybe do some further analysis later on down the line. So that would already help with that. Of course, if you want to, those individual images are always there that you can take them to third-party software. You can do some more advanced analysis if you wish. But because we are doing the analysis on the projected image, you can have the option to delete those immediately within a post run. So this will already save you in some data.
Excellent. Thank you very much for that answer. I'm afraid I'm going to have to wrap it up there today, but thank you again to Roman and Linda for today's informative discussion and presentation. Thank you to everyone joining us online and for your interesting questions. I hope that you have found this a worthwhile session. And I know if we have not got around to your question, we will definitely look into following these up by email. And if you do have any other questions, please feel free to email me at editor@selectscience.net, and I will follow up with your questions for our speakers. And just a note, there are additional resources for this webinar I would like to refer you to. You can find these in the resources tab to the left of your screen. This is where you can also find a certificate of attendance that you can download. So that is in the related resources tab to the left of your screen. If you would like to listen again to today's webinar or invite a friend to listen, it will be available to watch on demand in just a few days' time. Goodbye, and thank you once again for joining us.
Thank you.
Thank you.